Hello, everyone. This is our final presentation for the Data Science Clinic. We were part of the Center for Living Systems. So as you can see, we were part of the Center for Living Systems. Leah Ding was our external mentor. She's a data scientist for the DSI and Gardel lab. Our team members were Eve, Jared, Kruthi, and Victor. A quick overview of our project is we were testing AI models to verify performance of cell segmentation on labeled and label-free public datasets. We analyzed performance and compare results against ground truth and original paper, and the original paper was a systemic evaluation of computation methods for cell segmentation. So our mini problem is we're evaluating and subsequently improving the efficacy of models for cell segmentation. This allows for better cell segmentation and identification. It's primarily a modeling problem, and we analyze CellPose and Stardust models working with two datasets, TissueNet and LiveCell. So you can see in this left column, this is the Stardust, and then on the right two columns, we have CellPose and CellPose with TissueNet, and these plots right here are intersection over union, which we'll discuss in a little bit. So the data science problem, the main data science problem is of image classification, which is a supervised problem. There were two data sets, TissueNet, which is labeled, and LiveCell, which is unlabeled. We have used two different libraries, CellPose and Stardust, for our image classification attempts. For this problem, we will be utilizing intersection over union, recall, and precision to evaluate our predictions. So we talked a lot about intersection over union. This is a metric that is the degree of overlap between a predicted bounding box or mask and the corresponding ground truth bounding box or mask. The equation is the area of overlap, which is the intersection, over the area of union. Precision tells us how accurate the predictions are in terms of positive predictions, and the recall is how well the model detects the actual ground truth objects. So there's true positives, and these are predictions that match the ground truth objects with an intersection over union greater than some threshold t. There's false positives, in which predictions that do not have a matching ground truth object are predicted with IOU greater than the threshold. And these are false negatives, in which there's no prediction, but there should be one because it's falsely negatively predicted.
Okay, so throughout our project, we faced a couple of big challenges. The first one involved the inversion of masks for TissueNet images. So basically, one of our models, Stardust, doesn't have built-in two-channel model performance. So we tried to invert images to try and replicate that, but unfortunately, it did not work. The second big challenge involved ground truth reconstruction for live cell data, since this data set did not have really easily accessible annotations. So more on the inversion of images problem. For the Stardust model, since it didn't have built-in two-channel image prediction, we had to try and invert the images to try and replicate this. And so what do we mean by two channels? So in the TissueNet dataset, their images come in two different channels. The first channel involves a nuclei, and the second channel involves a membrane. So we try to invert the channel 1 images to try and replicate channel 0, but this process actually led to our model making many false positives, so we actually had to scrap this.
Our next major problem had to do with the live cell dataset. The live cell dataset contained the ground truths for our data, but it was in a form that was not easily accessible or useful for our models, and instead just as annotations. So on the left here, we can see the original image, and what we had to do in order to use the annotations for our actual predictions to get the metrics for our performance is we reconstructed the ground truths. And this was done using NumPy and a couple of other Python libraries to get the masks overall. So now moving on to our results. First, we have our results on the TissueNet data. This is both for Stardust and CellPose, and these two plots here are our IOU plots so the intersection over union. So on the left here, you could see that TissueNet does fairly well with the mean IOU scores across the images. And on the right, Stardust also performs similarly. So we see pretty much equal performance on TissueNet overall.
And now looking at the STARDIST results for the TissueNet data, we see that across the standard model and the versatile model, there's the best performance on lung and tonsil tissue samples, and the worst performance, quite poor performance on the spleen tissue. And we see generally that for the versatile model and the standard model, that the precision tends to be higher than the recall. And moving to the next figure, we can look at the cell pose results for the TissueNet data. And overall, there was better performance here than the STARDIST models. And again, better performance on lung tissue, and in this case, pancreas tissue. But as was the case with the STARDIST results, poor performance on spleen tissue. And we can see this in a sort of nicer visualization in a table. This next figure, where we've organized it by tissue type for the STARDIST models and then for cell pose. And we can see very clearly here that for the spleen tissues, both STARDIST and cell pose perform quite poorly. Why this might be, we can explore in further iterations of this project, looking to data quality, and perhaps the microscopy images were just blurry or had poor resolution or things like that, and why the models perform quite well on, for example, lung tissue.
Here we also have a figure for our recall and precision on the live cell data set. So overall here, we can see that the performance is quite poor again. And this is generally most likely because of image quality, and also perhaps because of the hyperparameter model tuning that we will do in the future. Um, so we could see that the scores overall are quite quite poor, but especially for one of the cell lines here. So again, this would be something that we would address in the future, potentially by looking through those images and tuning the model to perform better overall for all of our data set.
Okay, so to further make sure that what we are recall and precision results were accurately capturing the original cells in the original image, we also created some quality control plots to further visualize this. So here we see 3 rows of images in 3 different columns. The first column is the original image. The second column are the labels that we created using the STARDIST model Versatile. And the third column are the ground truths that we obtained from the TissueNet metadata. So as you can see in the top row, this is an example of a considered low accuracy label, and at the bottom we see an example of a high accuracy label. Some interesting thing to note here is that for our on the bottom row, we actually have a precision of 1, which means that there were no false positives in this image. So this is just an interesting example to note what our segmentation labels look like in an instance where precision is equal to 1.
Here we also have some similar plots for the live cell data set. You can see on the top is the lower accuracy and on the bottom is the higher accuracy. You can see also that the original images are quite poor quality, as we mentioned before, where the cells are not very visible, and not clearly defined. So it's possible that our model had trouble picking up on those specific areas because of the lack of contrast between the background and the cell itself. And in the middle, again, we have the labels, and then on the right, we have the ground truths. So you could see that the recall and the precision is quite low, and with our highest precision on the bottom being 0.72. And overall, there's a lot of cases where the model was not able to pick up on certain cells, likely because of image quality and, again, some tuning that we could do in the future to perhaps fix that.
So now just for a quick overview of our results, the cell pose model that we use had greater overall accuracy for recall and precision than both the STARDIST models on both the live cell and TissueNet datasets. The live cell dataset evaluation also had a high false negative rate, likely due to image quality. And both models performed best on the TissueNet data. The lung, pancreas, and tonsil cells had the highest accuracy, while the spleen cells had the lowest accuracy. However, as we did not show any live cell STARDIST model performance, we don't know if this also will stand true for that.
And then just wrapping up with conclusions and next steps for this project. Conclusions, we found that CellPose model has a higher accuracy than the Stardust model on public data sets, that is TissueNet and LiveCell, and that the image quality of the TissueNet data is much better than LiveCell, which may have contributed to those results. Moving forward, we're going to look at addressing the image quality issues of the LiveCell data set, and considering the accuracy differences between different cell types on the TissueNet data. Looking forward to hyperparameter tuning and training for increased accuracy. And then lastly, extending this project into analysis of 3D cell segmentation. And with that, that was the CLS project for autumn 2024. Thank you.